So what is a gene? You know what is a gene? A gene is nothing but it is a basic physical and functional unit of heredity, as you know. And here the genes, it consists of a DNA that gets transcribed and gets translated by a cell to make a proteins. And these structured proteins, these genes acts as instructors to make a molecules called proteins. And these proteins are they create invisible phenotypes. So the proteins ultimately, after translation, once it undergoes translation, the expression of these proteins, once a protein is formed, the expression of these proteins has a very great impact on what a gene has done, what is a gene, and how far the gene has been expressed, so on. So a lot of scientists they have worked on the gene as an heritability, as a molecule which is responsible for heritability, like Mendel, John Gregor Mendel, right from 1860s to 1900, a lot of work has been done. And in 1900, the rediscovery of Mendel work was done by Carl Corenson, Teschmark, and Hugo de Vries. They prompted the foundation of genetics, actually. So genes are the building blocks. As you know, they are the building blocks of the genes are consist of building blocks in the form of alphabets, what we call it as ATGC, that is adenine, guanine, thymine, and cytosine. So the the structure of the DNA, once the DNA structure was identified or it was framed by the biologist, the chemist, and the chemist and physicist, like it gave a compactness with the chemistry. So DNA is nothing but it is composed of a chemical genetics, chemistry, and as well as the biology, wherein a lots of people they worked on it. Next. So gene is encapsulated in chromatin structure, and you know that a chromatin is made up of the histone protein molecules, which are much more important to say that how they work on. So gene has a distinct locus. Each and every gene has its own locus or a own specific sites to express themselves. And the gene, right from 1900 to 1928, a lot of work was done with by Thomas Hunt Morgan and others, to say that how the genes are present on a definite position and they have been localized at a specific locus on a chromosome. So genes are made up, the genes consist of the molecules, like a gene as a physical molecule, and it consists of a protein, what we call it as a blueprint. And with the Hershey-Chase experiments, like it determined that DNA is consists of a genetic molecule, genetic material. So coming to history of DNA, the experiments with reference to Griffith were in transforming principle experiments, X-ray photographic studies of DNA structure, and Hershey-Chase experiments for to constitute the DNA constitute the genetic material. All this gave rise to the birth of chemistry. So gene has its own codes. It has its own code, and that is in the form of four bases A, AT, and GC. The concept of gene is based on the genotype and phenotype, nucleotide sequences, structure, and function. So many scientists right from 1866, Gregor John Mendel, Gerard, Beadle and Tatum, Ingram, and Ionovsky, they worked on the different concepts of the gene in the form of like a unit factor that controls specific phenotypic traits. A gene is nothing but one gene, one metabolic block theory was studied by Gerard. Then came one gene, one enzyme theory, followed by one gene polypeptide theory. So the gene has been framed into a language of genetics in the form of once the Mendelian concept was proved, experiments were done, the principles of Mendelian experiments, it gave rise to a lot of language, like the codes that gave rise to a lot of languages, the recessiveness, the traits like the phenotype, alleles, then heterozygous, homozygous, genotypes, phenotypes, genes, so on. So the gene has a fine structure. So gene has a fine structure right from promoter region to terminator region, right from it reads from 5' to 3' end. And in between, it constitutes a lot of information like the promoter region, which consists of the transcription factor binding site like Tata box, followed by the UTR region. Then comes the transcription start site, and then it is interspersed with exons and introns, wherein then it ultimately ends up with the downstream region, that is at 3' region. So gene structure is the organization of a specialized sequence elements within a gene. So this is the timeline discovery of the gene, right from 1800s. We have a lot of information with reference to the gene, and now we are at the here you can see 1800s. It started from 1800s, and it is in 1865 the Mendelian principles, followed by the isolation of DNA, chromosome theory of hereditary, DNA as a semi-conservative DNA as a semi-conservative molecule, then X-ray diffraction studies of a DNA genetic code. All these were unraveled, and it gave rise to like in 2002, we have the microRNA in cancer, the role of microRNAs in cancer, transcription factors in 2007. Likewise, a lot of things have been discovered, and it has now we are in the era of the histone acetylases, histone deacetylases, DNA methyl transferases, transcription factors, so on. So Mendelism is mainly focused on the pre-Mendel hypothesis of hereditary, Mendelian concept, post-Mendelian concept. So what is a pre-Mendelian concept? Pre-Mendelian concept is also known as the preformationism, wherein it is suggested that organisms were created at the same time, and the succeeding generations grow from homunculi or animalcules that have existed since the beginning of the creation. So next came the Mendelian concept, wherein in Mendelian concept, the genes was invisible and treated as statistical entity. The pattern of inheritance was deduced through ratios like 3:1 ratios, or 1:2, 1:2:1 ratio, or 9:3:1 ratio. Then came post-Mendelian concept, that is the recapitulation of phenotype and genotype, which has revealed based on the dynamics of Mendelian inheritance. And the basic petals of Mendelism is the factors or a trait or a character of a gene. So post-1900 investigation revealed many facets of inheritance as visualized by Mendel, namely incomplete dominance or codominance, interaction of genes, sex linkage, crossing over, genome imprinting, and epigenetics. So here, the genotype to phenotype. So genes have given rise right from genotype to phenotype. And what is the role of these genes? So how the genotype has been framed? So for which William Johansen in 1909, he introduced the phenotype and genotype in the Dictionary of the Genetics. And as you all know, a phenotype is the appearance of an organism, wherein the genotype is the genetic constituent of an organism. So how this phenotype is brought about? Like there is a lot of variations we do come across. And because of the variation, a lot many phenotypes have been observed. And these mutations, and these are brought about through the mutations. So mutations, they are the heritable changes in genetic information that they change the DNA sequences. It may be the chromosomal mutation, it may be the gene mutations. Ultimately, mutation is the catalyst for evolution. So unless and until if there was no mutation, then a variation would not have been existed, and we could have not come across the phenotypes, variations or variants in the system. So either it is a loss of function mutation or a gain of function mutation. It relies on the impact on protein and its fitness. So the depth of the mutation, it depends on the impact of variants. It may be the level of penetrance of the mutation, the strength of the genotype or a phenotype, then the timing of impact of the mutations. All this, at the end, it gives rise to the phenotype. It modulates all this performance put together, and it ends up with a phenotype. So genetics is the science of hereditary, and genetics deals with the molecular function of genes and gene behavior in the context of a cell or organism patterns of inheritance from parents to offspring. and gene distribution, variation, and change in population, such as through genome. So, progress is that how do genes or DNA determine different characters or a phenotype? And they have to undergo a lot of transaction. Gene transaction has to undergo, and gene transaction it undergoes through and through a step of through a stepwise, like it has to undergo a replication, transcription, recombination, regulation. On the other hand, while during this regulation of gene transaction, the mutations, the inbuilt mechanism is there, like how it repairs the mutations that happens, and all these they have to undergo regulations. And once the regulations do takes place, the genotype of a DNA will give rise to a phenotype. That is the protein which with reference to a phenotype, which ends up with a phenotype. So these are the some scientists who worked on it since from 1865. So then comes the genome imprinting. Genome imprinting is the phenomenon of parent of origin or gene expression, wherein the genes that undergo genomic imprinting, the parent of origin is often marked or stamped on the gene during the formation of egg and sperm cells. This stamping process is called methylation and is a chemical reaction that attaches small molecules called methyl groups to certain segments of DNA. So the difference between genetics and epigenetics is that the traditional genetics, it describes the way the DNA sequences in our genes and are passed on from generation to the next, while epigenetics describes the passing on the way the genes are used. So here, the epigenetic regulation of gene expression is a common process that acts during the differentiation of somatic cells as well as in response to environmental cues and stressors, and passing on these modulations to the offspring, which constitutes the epigenetic inheritance. So what is epigenesis then? So here, the germ cells consist of homogeneous substances. The parts of an organism arise progressively, and development was thus viewed as a process of new formation from which the epigenesis is derived. Here, right from the cells has to undergo a lot of morphogenetic events to have a topology of the tissues, and thereby this has to undergo a gene regulation, ultimately to form a genome. On the other hand, the gene has to get expressed to form the division, migration, or addition of the cells. So thereby, epigenetics theory of epigenesis was formulated by German physician and naturalist F. Wolf. He claimed that the structures arise during development that are not already preformed. So later on, that in 1700 itself, the epigenesis was known, and later on, Conrad Waddington in 1942 defined the term epigenetics. It is the study of how behaviors and environment can cause changes that affect the way your genes work. So unlike the genetic changes, epigenetic changes are reversible and do not change your DNA sequence, but they can change how your body reads a DNA sequence in the form of phenotype. So at each and every stages of development, a lot of alterations do happen, like they have to undergo fertilization, then followed by cleavage, gastrulation, organogenesis, or growth, then leads to new phenotype. So at each and every stages of development, the genes has to undergo switch and on mechanisms. So these are the sequential breakthrough of gene to genomes. Then the genome, coming to genome, the genome is the entire genetic complement of a cell, wherein your genome is one whole set of all your genes plus all the DNA between your genes. So major types of genomes are like genome changes takes place through base substitution. It may be through the copy number variations or SNPs. So through all this, the rearrangement of the genes do happen. So the complexity of the genome size depends, the complexity of the genome depends upon the size of the genome. Here, right from the viruses to plants, a lot of complexity do arise with the increasing complexity. So we have the Homo sapiens, the humans, they have the chromosome number of 46, which is the estimated genome size is about 290 into 10 to the power of 6. Estimated gene number is about 30,000, and estimated gene density is about 1,000, 1 in 100 base pairs. So likewise, the smallest Drosophila melanogaster, if we take on this, like it has a small set of chromosomes, that is 8, and estimated genome size is about 180 into 10 to the power of 5, 30 with 13,600 estimated genes, and 1 in 9,000 genes per base pairs are present. So, whatever it may be, the genome structural integrity relies upon the chromosomal stability. So, one has to have the stability of the chromosome should be there to have a structural integrity of the genome. A genome dynamics should happen at a given space, spatiotemporal pattern. Or DNA repair and enzymology is also equivalently important, and mechanism of mutations are also much more important. And apart from this, we have the fidelity of the replication is also at most important to have the genome structural integrity. So history of the gene, it way backs to gives in 1900. The DNA was when the DNA was first identified as important for life. It gives rise to the developing new sequencing technology to understand genomic function, to analyze variations in human genome sequences, to address ethical, legal, social issues related to genomics, and advancing the field of bioinformatics and computational biology. And now we are in the era of artificial intelligence and the machine learning. So the study of genome is a discipline to unravel the genetic content of the genome by way of sequencing that is determined the exact nucleotide sequence of the genome, wherein the appearance of the genome, it is the phenotype, it is an interaction that takes place through genes and the environmental factors. So genomics, the term was first introduced by Roderick in 1986. How the entire genome of a species function as a unit and evolves over time through variations with sequences. It is the life's blueprint, life diversity, and life history. It involves all gene at DNA, RNA, and proteomic level, as well as tissue and cellular level. To analyze, the analysis of genome content is reliable on ultimately overall the analysis of the genome. It relies on the bioinformatics. So there are many sequences that aroused, right? Like first generation sequencing, we have second generation. So at each and every sequencing technology, it has been, it has given rise to a lot of modulations with the first generation sequencing, then a little bit more advanced with the second generation sequencing. We have the Illumina sequencing platform that has emerged as the most successful technology to the point of nearby monopoly, and Illumina success can be attributed to its high throughput capabilities, accuracy, and low cost per base, followed by PacBio sequencing, then Oxford Nanopore sequencing, Ion Torrent sequencing. All this sequencing technology has led to polished DNA sequencing within a short time. And so there are four different forms of genome, types of genomes. One is structural genomics, functional genomics, followed by comparative genomics and mutational genomics. So coming to structural genomics, what is structural genomics? Actually, it determines the structure of every protein that has encoded by a genome. That is what we call it as a structure. So coming to functional genomics, it aims to collect and use data from sequencing for describing gene and protein functions, followed by comparative genomics that aims to compile the genomic features between more than one or between more than two different species. Then coming to mutational genomics is the one that determines the mutation that occur in a person's DNA or a genome. So either it is a structural genomics or a functional genomics or comparative genomics or mutational genomics. It relies upon the integration of the genetic content, gene activity, and protein function. So the total attribution of this gene, structural, functional, and comparative and mutational genetics, ultimately it ends up with the gene content, gene activity, and protein activity in general. So structural genomics accelerates the diverse areas of biomedical research, which is at most important to have an early structure in biomedical and functional genomics programs, to have the novel technologies which accelerate the traditional focused structural biology. And also structural genomics accelerates the structure-based drug designing processes and protein production technologies. So we have the genome transcript coming to transcriptomics. Like it has to undergo through the genome. We have the genome. It has to undergo expression. Once it gets expressed, like with the different sets of proteins are produced, and this what we call it as a knowledge of protein, like it is a proteome, and each of these proteins, they have their own metabolomics, and it ends up with the metabolome. So right from DNA transcriptome followed by proteome. Here in the transcriptome, right from RNA to mRNA, alternative splicing mRNA editing will takes place, and then polyadenylation do happen. And here in the proteins, a lot of modification of the proteins to takes place, and ultimately it ends up with a biological function, what you see here. Then comparative genomics is the one which compares comparison of one genome to another genome. Again, here also it includes the genomics followed by transcriptomics, proteomics. Here it is the functional genomics which operates on transcriptomics and the proteomics, and ultimately it ends up with the metabolomics, wherein here transcription followed by translation and enzymatic reactions are at most important to have the metabolites. So here the one-to-one comparison will be easier, right from humans, chimps, urchins, and mouse or Drosophila. Coming to mutational genomics characterizes the mutations which are associated with genes, and basically focuses on the genomic, epigenomic, and transcript alterations in cancer. And it bears similarity to genetical genomics with linkage genotype and transcriptional states. The difficult task in this genomics is the finding of genes that are underlying the transcriptional changes. So we have the mutational process wherein it undergoes through DNA replication, genotoxins, DNA repair deficiency, DNA editing of the somatic mutations. And this brings about the small mutations, structural variants, and copy number variations. And thereby they get translocated or inversions or loss of function mutation or gain of function mutation. Might be that leads to the mutational signatures. And in turn, these mutational signatures, they give rise to the tumor sequencing, followed by genomic landscape, patterns of mutations, and overall the mechanism which brings about the variant calling catalog mutations, modeling, link pattern mutagenesis, so on. So history of genomics, it starts from 1950 and still it is a lot of things have been unraveled. Frederick Sanger, a pioneer of sequencing, Sanger is one of the few scientists who was awarded 2 Nobel Prizes, one for sequencing of the proteins and other for sequencing of the DNA. So comes the mapping. Here the mapping have advanced in sequence-based error, close interplay of mapping and sequencing in the exploration of genomes. It gives rise to the availability of back libraries in allowing physical mapping of more species of the genomes. So here the genomics, it includes the sample preparation. We need to have the DNA amplification followed by PCR purification, then sequencing pre-preparation, DNA sequencing. And here, this has to, in case of DNA amplification, once it reaches the DNA amplification, here the molecules which are used are, it may be the DNTPs, DNA forward primers, reverse primers, so on. And then here at this stage, we need to have the denaturation labeling of DNTPs. So here, at the sample preparation stage itself, the proteinase K is important to denature the protein content. And here, the spin volume column DNA extracting method, spin column based PCR purification. So this is the Sanger sequencing technique, which has been more used, and I won't go much into it. So DNA double-stranded denaturation has to take place, followed by make multiple copies of a segment of a DNA, then attaches primer has to be attached and to 4 polymerase, and then followed by polymerase chain reactions, followed by grow complementary chains until termination denaturation of the grown chains and electrophoresis for the 4 molecules. So this is the fluorescent DNA technology, wherein the primer for replication has been used, and here the strand these are the strands that has to be sequenced. Then we need to have the preparation of 4 reaction mixtures, and all this will include the enzymatic reaction that takes place to have the target DNA sequencing, followed by single-stranded DNA sequencing. Then labeled complementary probe has been labeled here. Then primary antibodies gets added on, and thereby secondary antibodies get conjugated, and then ultimately primers and fluorescent probes are made. So the whole genome followed by whole genome sequencing, it is nothing but the genomic DNA we have. It has to be undergone fragmentation or else the sharing of the DNA. Then it will be processed with the next generation sequencing, and ultimately it ends up with the data analysis. So there are many sets of genome manipulation techniques are there, and they are very much cumbersome. With the knockout, the genomic manipulation could be done for knockout, or editing could be done, or interference or activation of CRISPR could be done. So in order to have a knockout, like disruption of function of for a specific gene can result from many different modifications. To edit, execution of specific sequences are required for the modification at a specific genomic locations. So unnotted genomic entity is read through the data banks, and DNA segments contribute to a phenotype or a function. In the absence of demonstrated function, a gene may be characterized by sequence and transcription or homology. There are gene expression signatures that has been which defines a phenotype such as cell and tissue state. And here, the combining protein structure features with a GES significantly enhances the reliability of transcriptomic signatures across the data sets and represents the various phenotypes, including the relationship among tissues and the drugs. So here, genome function relies upon the the recombination, that is histone protein histone protein transcription translational modifications, which takes place during the recombination or during the repair mechanism or transcription and replication. So in recombination, it is the genetic diversity or meiotic recombination that do happen, which results in the chromatin, it may result in the chromatin modification. And here at the transcriptome level, it is the gene activation, gene repression, transcriptional regulation, chromatin accessibility. Then it is followed by replication, wherein the DNA replication, timing is important, replication origin setup, genome stability, and chromatin structure. Whereas in case of the DNA repair, it is the DNA damage responses are important, followed by double strand breaks, homologous recombination, and non-homologous and joining. So applications of genome editing, it involves the three main structures like phenomena like medicine. It is involved in the biology and biotechnology. Whereas the medicine, again, it develops the drug development. For the drug development, and also it is involved in gene surgery. Whereas in biology, various animal models to be used and genetic to see the genetic variations. At biotechnologic level, the food, biofuel, and other materials are very much required for the applications of the genome editing. So there are different forms of writers and errors, what we call it as like enzymology. If to edit a gene or to the faith, we say that we have each and every organism, they have their own phenotype, they have their own signatures, and these signatures could be altered by having the genome editing units like writers and erasers. And there are different forms of writers and erasers, especially with the meganucleases, zinc finger nucleases, TALEN, and CRISPR-R. So all these molecules, the gene of interest could be altered and the faith could be written. Here we have the epigenetic writers. These epigenetic writers, they can also go through the epigenetic reader. Epigenetic writers are in the sense the methylases, acylases, which undergoes the epigenetic erasers. And in turn, these epigenetic erasers, they may lead to the changes in DNA damage repair, changes in DNA replication, or the changes that has happened during the transcriptional activation or repression. So all these molecules are very important to have the genome editing, like HDACs, the bromodomains, acetyls, methylases, acylases, methylases, phosphates, so on. So CRISPR-RNA is the most efficient technology or a tool to have the genome editing. So efficient kit, it is Cas9 is an efficient kit for genome manipulations to bring about the base editing, to have an RNA editing, to have the imaging, transcription, epigenome, so on. In turn, all this like RNA imaging, transcript knockout, pathogen identification, genotyping, DNA barcoding, all this could be accessed through this CAST toolkit. So functional genomics has impact on for example, in fruit trees, like the whole genome variations, genetics and breeding, linkage map, duplication and transcription factors, gene expression based on biomarkers, transcriptomics and metabolomics, and all these are brought about through the functional genomics. And at the same time, there are a lot of challenges are there with reference to massive genome sequence coupled with the functional genomics and system biology with reference to ecosystem analysis, modification, evolution and classification, medical discovery, biofuel development, crop improvement, so on. So molecular genomics is a critical component of expanding database linking alterations of DNA and RNA with diseases, disease prognosis, and therapeutic responses, which are been structured with the component compactness of omics. So as you all know, the genomics followed by proteomics, then metabolomics and bioinformatics. All this, either it is a genomic or a proteomics or metabolomics or a proteomics, all these are read or through the bioinformatics. Genomic is read as a molecular genome, and all this put together, it is a molecular genomics. And here it is a characterization of genome function on protein level, or recombinant gene expression do happens, whereas at the genomic level, the genome and gene structure characterization to happen with the DNA sequence analysis. It includes gene expression analysis, gene and promoter field cloning and characterization and molecular markers. Followed by metabolomics, it is the metabolic profile and status analysis, identification and characterization of metabolite synthesis and accumulation. So all this information is read through biology, what we call it as the bioinformatics. That is a data set, through the data sets. So coming to the introduction of omics, we have the genomics, transcriptomics, proteomics, and metabolomics. And here it is the genomics, it reads through the cognitive, comparative, functional, and metagenomic. It is used for the neuromics, personal genomics, and epigenomics studies. Transcriptomics, it is a protein at a transcriptional level. Proteomics to study the immunoproteomics, nutriproteomics, proteogenomics, structural genomics, so on. Metabolomics relies upon the metabolomics and metabonomics. So ultimately, it is a genome, transcriptome, or proteome, or metabolome. It ends up with the metabolite like this, and at the end, we have a functional phenotype that leads to a functional phenotype. So coming to the insights in the molecular genomics, the omics is based on the system, like a five system, that is epigenome or a genome, epitranscriptome or an epitranscriptome, with a normal set of DNA and the epigenome. This is with reference epidNA or a gene with reference to. Then protein with our epiprotein or proteome, and metabolite with the metabolome, lipidome, fluxomics, and phenotype, or ultimately it leads with a phenotype or a phenome or exposome. So all these are together. When we say that it is an epigenome or an epigenetics, then it ultimately it relies upon the environmental cues also, along with the genetic structure. So high throughput data is analyzed through computational tools. Bioinformatics is required. In silico screening will be done. Pathway signaling, interaction, and network modeling. So that deals with the molecular genomics. So multi-omics variations, how the multi-omics model to say that we have the epigenome, which gives rise to the genome, that is a chromosomal rearrangement, and at the genetic mutations level. Here, epigenome is the one wherein we study at the DNA methylation, which leads to the histone modification. And we have the transcriptome, that is the fusion transcript, which leads to the alternative splicing. So all this together, like it in the invasion, metastasis, and angiogenesis, and here it leads on the other hand, evasion of immune cells apoptosis, it leads to the cancer genetics. So here, whereby the integration of molecular genetic data is structured on the omics is structured by the integration of the molecular genetic data. So how it is imbibed is that the genomics, that we need to have the genomic data. On the other hand, we need to have the epigenomic data here at the transcriptome level, proteomic level, metabolomic level, and phenomics level. And they have to get organized. The organization has to be there in the form of cells, organelle, organs, or organisms. So it ultimately, all these factors, it depends upon the age and as well as the environment. So this is about the proteogenomics, wherein we need to have, for proteogenomics, we need to have the genomics, wherein the whole genome sequence, whole exome sequence should be have, and that through which a multi-omic field, that is the proteogenomics is arrived, and then comes the proteomics and transcriptomics. So proteomics is nothing but it is a protein complement expressed by a genome of cell is a protein of a cell is a proteome. Proteomes are much bigger than genomes, one genome, but many proteomes. It is proteolytic processing that is the temporal and developmental regulation of protein synthesis or degradation. Through degradation, it happens and emerges with a tissue specific expression or at cellular localization. So, post translation modification do happen through phosphorylation or acetylation. These are the tools that are used to analyze the proteomics, that is MS based methods, then affinity reagent array based on the protein microarrays, antibody microarrays, and tissue microarrays, so on. So there are different avenues of proteomics, like leads to protein expression profiling on one hand. On the other hand, it has this protein expression profiling. It gives rise to the to know the medical microbiology, to have a signal transduction, disease mechanism, how the disease do occur, and post translation modification, and also it brings about the functional proteomics. And on the other side, it has a structural proteomics, which deals with the organelle composition, subproteome isolation, and protein complexes. Metabolomics is the systematic study of metabolites in an organism and how they change in relation to our biological perturbations, right from the genomics to genomics genomics to translation, transcriptomics, proteomics through transcription, translation, enzymatic reactions, and ultimately right from genotype to it ends up from right from genotype to phenotype. So the collection and integration of multi-omic data, it relies upon the genome. So this is just an example to show like a collection and integration of multi-omic data is important, followed by for multi-omics analysis of data by system genomics, followed by results and validation in system genomics, and the applicative part of it with the animal health, production, and welfare. So authentication of the genomics depends upon ultimately the authentication of genomics depends upon the database integration, mining, visualization, and annotation, which has its own network. They have their own pathways and they have their own modeling, which brings through the gene pathway and genome function that gives rise to the genetic perturbation. On the other hand, this genetic perturbation gives rise to a phenomics, that is what we call it as a phenotype, which ends up with a phenotype, and here the genome sequencing, RNA sequencing, proteomics, as usual, it goes on. So there is a conceptual structure of genomic portals which has been functioning. You need to have the genome data that is in the form of gene expression, transcription factor binding, that is CPG pools, that is islands, and gene ontology. One has to know the interaction of the networks, and functional knowledge base is also important on the other hand to have the genomic portals with the literature concept and transcription modulations. And we need to have the statistical analysis, and also machine learning is also very important to have a genomic portal and the visualization on the and so wherein we can achieve the new functional knowledge, new physiological understanding, and new testable hypothesis for this is the conceptual structure to go for the genomic portals. So here the genome manipulation techniques, which involves the biochemistry, the biochemistry, its function leads to the genetics, genes, molecular biology, and proteins all to put together. It has its own the like the basic molecular biology techniques, which involves the genomics techniques, that is sequencing genomes. It is then proteins. All these techniques are very much required to have a genome manipulation. So discoveries from DNA to genes, genes to genomics, genome and genomics to, and now we are in the genomics to era of medicine. So right from the gene, we have enriched the genes to genetics, genetics to genome, genome to genomic era, wherein this genomic era has enhanced the genomic era of the medicines, and for which throughput information, like a lot of work has been done since from the human genome project, which has completed in 2003. It has given rise to many information with regard to the era of genomics. So advances in genomics has led to bioinformatics. and through which one can know the DNA methylation, chromatin modification at the nucleosome level, DNA methylation at the histone level, DNA hypersensitive sites to read the functional genomic elements, and as such transcription factors which are binding, and the long range transcribed region could be identified through these bioinformatic techniques. So genomics is relevant to practice the precision preconception and prenatal testings, to have the newborn screening, disease susceptibility, screening and diagnosis, prognosis and therapeutic decisions, monitoring disease burden and recurrence. So genomics has led to the relevant practice. So genomics is one of the important, it is an which has made an history to learn how properly the functions, the genome functions at different stages of development. So the core, so for which we need to have the knowledge of core genetics. So right away, right from gene, we can't have a roadmap to the genome or a genomic era. But here we need to understand the core genetic knowledge through reflecting practices, identifying the patients, ongoing nursing care is also important, and communicating genetic information which is much more important to have the knowledge on the. So here bioinformatics, present and future, it relies upon the primary database containing large-scale experimental data in genomics and proteomics, and it leads to the understanding of functions and utilities of individual genes or proteins, followed by the secondary databases containing accumulated biological knowledge to understand the functions and utilities at the molecular level. This is at the primary database at the individual level, secondary database at the molecular level, at the understanding the function and utilities at the molecular, cellular, and organism level. That is through the bioinformatics. Now, what we have then a complete computer representation of the cell and organism is the bioinformatics in future to understand the basic principles of higher complexity of biological system. That is what we are stepped into the era of artificial intelligence that reads, that writes, that describes, and that assign a particular assign a particular medicine that has to be there for the misexpression of the genes that has taken place with. So bioinformatics, it relies upon the molecular biology, it relies upon the information technology, it manages in the management of the informations, applicative databases, computational resources. Then we have the genomic, proteomics, and overall the omics that is added upon. And here the computational that has led to the computational molecular biology with all this information with regard to genomics, bioinformatics, structural genomics, and proteomics. And in general, this computational biology, molecular biology, which reads through the computational biology, and now it ends up with a machine learning. So to have a genome data, we need to have the hypothesis driven through functional testing. That is, how do mutations affect cell behavior? How do mutations contribute disease phenotype? The approaches to understand cell differentiation, pluripotency, and cancer cells, so on. So we need to have the to integrate into knowledge network. We need to have inform the selection of therapeutic lines, and clinical pharma studies of cancer are much more required. So regarding the global screening strategies and techniques driving omics, as you all know, like we have the genomics, proteomics, epigenomics, and metabolomics. Here, the genomics is the one wherein we screen through the mRNA, microRNA, microRNA, microarrays, non-coding RNA microarrays, RNA sequencing, whereas proteomics with reference to expression of proteomics, structural proteomics, functional proteomics, and function. Wherein metabolomics, it's read through metabolite profiling, metabolite fingerprinting, and epigenome, it is read through the DNA methylation profiling, chip sequences, genome sequences, exome capture, and RNA sequencing techniques. So to analyze the genome data, like we need to have a patient with technologies, a well-built technologies now we have, and these technologies should be used to have a good analysis, data analysis For example, for genomics, we need to have the point mutation, small indels, copy number variations, structural variations. Likewise, for transcriptomics, we need to have the differential expression. We'll come to know about the gene fusion, alternative splicing, RNA editing, and for epigenomic, we need to have the methylation, histone modification. We need to know about the transcription factor binding sites. So all this will have an effect on the integration and interpretation of all this in the form of functional effect of mutation, network and pathway analysis, and integrative analysis. Further, it leads to the understanding of cancer and clinical applications. So applications of genomics, like it is the genetic paternity test, followed by genetic compatibility test, followed by the genetic overall to have a genetic fingerprint, and to have the genetic ancestry we need to know about the genetic disease risk, and once the genetic disease risk is found out, then we can have the personalized medicine that could be framed. Coming to genomics and public health, human disease results from gene and environmental interaction. So public health leadership needed to translate gene discoveries. So genetic affects all the public health functions in the form of assessment, policy development, and assurance. Public health must plan to train the workforce in order to build genetics capacity across programs and the knowledge generations, population sciences, genome-based science technology, humanities, and social. So it has a lot of societal impact. So we have the Human Genome Project that has established, ultimately the Human Genome Project is the one which has established a fertile foundation, and that has represented the culmination of an extensive foundation that has been laid before its initiation, with the likes of Mendel, Morgan, Sanger, Watson Crick, Rosalind Franklin, along with a few of inquisitive intellectuals behind the eventual elucidation of Enigma, the human genome. So the aim and objective of the Human Genome Project was ultimately to understand the functional genes, to make the information available for the researchers, to develop tools for processing, analyzing the data. So ultimately, the outcome of the Human Genome Project has given rise to characterization of diseases and genes. It has laid a foundation for system biology, bioinformatics, DNA sequencing, physical map of human chromosome, genetic variations, so on. Aftermath faced by the Human Genome Project, a predominantly practical and ethical opposition has been faced with the conception of the Human Genome Project with reference to type of opposition, that is ethical opposition, then practical opposition. The type of opposition that has led to the results and worried are the patenting genes, genetic discrimination, genetic enhancement, informed consistent, wherein here the practical opposition, that is big versus small science, funding favoritism, future of research, so on. So the current and the related events that has been led with these resulting worries have been cited here. So with the finish of Human Genome Project, like we have like in 2003, the Human Genome Project, which started in 1990 October and ended up with April 2001, 3. And here with this timeline, a lot of finished Human Genome Project have been mapping projects have been done with Cerevisiae, Trypanosoma cruzi, Arabidopsis thaliana, Neurospora crassa, and Pseudomonas putida. So the vision of genomics is that, so to have one has to have the new genomic knowledge, quality which leads to the quality improvement strategies, genomic medicine practices, innovation, outcome of the data collection, outcome of data analysis. Ultimately, it ends up with the genome learning healthcare system. On the other hand, we need to know about the basic genomic research, that is the genome biology hypothesis, followed by genome technology innovation, genomic data, and genomic data analysis.